Okay, so you have your double helix DNA strand, and every replication starts on a specific part of the DNA that is called the origin of replication. The first point in the replication phase is the enzyme helicase. This is then attached to the origin of replication. The helicase is used to separate the hydrogen bonds and strands of the DNA into two. But because the bonds are so strong, they try to form back together. To make sure the bonds don't bind together, single-strand binding proteins are used. So, single-strand binding proteins bind. They bind to a single complementary bond to prevent them from binding back together. All of this causes the formation of a replication bubble, a space in between the two separated strands of DNA. At the end of these bubbles are replication forks, Y-shaped regions where the parental strands of DNA are unwound. The enzyme primase is added to the site by the origin. This then creates an RNA primer that attaches to the other complementary strand. Just pretend. There. The initial nucleoid chain produces as a result DNA synthesis is a short strand of RNA rather than DNA. This chain is known as a primer and its synthesase primase, an enzyme that begins a complementary RNA chain from the single primer. The new completed primer is base paired to the template strand. The new DNA starts from the 3' end of the RNA primer. However, the two strands of DNA in the double helix are antiparallel, meaning the strands are oriented in opposite directions. There's a 3 and a 5 end of each strand of DNA. The 3 end can only be attached to the 5 end of DNA. The DNA strand going to the 3 and the 5 direction have to be attached to the RNA primer, or DNA strand going in the 5 to 3 direction. The difference between 3 to 5 and 5 to 3 is that the strands are going opposite directions. So one side is 3 and one side is 5, and then one side is 5, but one side is 3. After this process, the Okazaki fragments are created. So the Okazaki fragments go on that one where they are put in by fragments. The main non-broken strand is called the leading strand, so that would be the strand where it has one complete piece of Play-Doh. And the broken strand is called the lagging strand, and it is separated. And so this DNA is now a daughter. It's beautiful. Telomeres are used for the function of fusing neighboring chromosomes. They do not actually contain genes. They do include multiple repetitions of nucleoid excisions. Telomerase is a ribonucleoprotein that is an enzyme that adds DNA sequence repeats to the 3 end of the DNA strand in the telomere region. And yeah.